Next up, I'd like to welcome Grace Lim from TCE2.
Okay, hi everyone. My name is Grace, and the title of my talk is Aurora Kinase, a novel regulator of TERT in thyroid cancer. Patients with advanced metastatic radiation iodine refractory thyroid cancer have a 10% 10-year survival. Traditional chemotherapy regimens have little efficacy, and until recently, there were no clinical alternatives for these patients. Systematic therapies that are now available include lenvatinib or sorafenib, which are multi-kinase inhibitors, which modestly improve progression-free survival. However, their use is often confined by toxicity-related adverse events. Thus, there is an urgent need for new targeted treatments for these patient group to improve prognosis with minimal side effects. A hallmark of cancer cells is their unlimited replicative capacity afforded to them by their ability to sustain the length of telomeres. Reactivation of telomerase, the telomere repair enzyme, is achieved by the activation of normally silent TERT gene by non-coding mutations within the TERT promoter, found in a majority of cancer types, including melanoma, glioma, and thyroid cancer. TERT promoter mutations and their mechanism of actions are unique to cancer cells, making them attractive chemotherapeutic targets. And this is particularly true for thyroid cancers, where there is now considerable clinical evidence that presence of these TERT promoter mutations are related to poorer prognosis of disease. Our group sought to discover agents that outperform existing therapies in the inhibitory action upon mutant TERT promoter activity in thyroid cancer cells. We screened a library of 218 well-defined agents with a CRISPR-edited thyroid cancer cell line by Dr. Martin Bullock. And in this screen, we identified a family of kinases called Aurora kinases that was able to suppress TERT promoter activity. Aurora kinases are crucial for cell cycle with different roles for each member of the whole cell division. And in cell cycle, it has been found that it is overexpressed in thyroid tumors and immortalized cell lines. Thus, the aim of the project was to demonstrate that Aurora kinases are a regulator of TERT in thyroid cancer. We found that Aurora kinase inhibitor hesperadin suppressed TERT promoter activity with an IC50 of 0.2 micromolar concentration. Inhibition that also led to suppressed levels of the promoter activity in the wild-type or mutant TERT promoter expressing cells. TERT gene expression levels decreased following treatment by at least threefold relative to the vehicle control, which was prolonged for 72 hours, and telomerase activity decreased by more than 90% following treatment, which also was prolonged for 5 days in 3 different thyroid cancer cell lines. Through this project, we discovered that targeting Aurora kinases suppresses TERT promoter activity, gene expression, and telomerase activity. This is a novel discovery of Aurora kinases and their unknown regulatory action in thyroid cancer that can identify them as potential targets for treating mutant TERT promoter positive advanced types of thyroid cancer. This project was showcased at 2 international meetings and at 1 local. And I would like to thank Sydney Vital for the PhD scholarship, my 2 supervisors and collaborators at CMRI and our cancer genetics laboratory team. Thank you.